All right, welcome back to Burt's Basics, Box Elder High School, Medical Anatomy and Physiology. Today we are tackling the beast of active transport versus passive transport. So let's look at the things you need to know. So if we look at this list, there's kind of a big list here, and we're going to divide it into two groups. We're going to talk about passive transport. We're going to talk about active transport, and these are the categories we have to know. Now, these guys get a little confused. You always have to look at the diagrams to make sure you know you're going the right direction and know you're going the right gradient. I'll explain those as we go through this video, and then we'll talk about iso, hypo, hyper as we go through this video. So let's start by talking about passive versus active. Now, this might be the dumbest diagram you have ever seen, but it is definitely the easiest way to think about it. If you are passive transport, man, it is going downhill. It is natural. And look at that little guy up here. Up here, he's like, man, this is easy. Versus active transport, just in the word active, that's weird. And so in this case, he's pushing that rock up the hill. So now let's change this from the simple diagram to something that's a little more our style. In order to be passive transport, there's a couple rules you have to abide by. Rule number one, you cannot use energy. Remember, if the ball rolls downhill, it's generally going to do it on its own. So passive transport, there's no energy, there's no ATP. So right away, that's a red flag. If you see the word ATP, you got to roll with active transport because it's not passive. The second thing is, it says most test questions are going to talk about the word gradient. Gradient is a fancy word for a slope or a hill. So in passive transport, it always goes with its gradient or down its gradient. And the last thing is on passive transport, since it's just natural, it tends to move smaller particles. Now, if we take active transport, it's almost the exact opposite. So if we look, this requires energy. It's unnatural. It's not common. So you have to put energy into the system. It goes against its concentration gradient or up its concentration gradient. Remember, gradient's a fancy word for a hill or slope. So in passive transport, you go with your gradient. Active transport, you go against your gradient. And if I'm going to spend energy, I ain't moving one particle. I'm moving a ton. So in active transport, we tend to move millions of particles, a big bulk bunch of particles. So what we're going to do is now we're going to look at each form of passive really quick. So remember, this is going to be pretty quick going through each one of these. You guys have your detailed notes online. So if you guys want to read your detailed notes as I talk about them, you're more than welcome to. Let's start with the almighty diffusion. Now remember, this is a passive transport. All of these are passive transports. In order to go passive transport, you must go from high to low or down your gradient. So if we look, here's time at the bottom. You're starting with a ton of people on the outside. They don't like this. They want to disperse and go from high to low. There's nobody over here, so this is very low. So over time, they will start to go through the cell membrane until they are evenly distributed. That is called diffusion. So a lot of things diffuse, and it depends really on what particle you are. In this case, these are all phospholipids, so anything that can diffuse can go right through my lipid bilayer. In contrast, look at the next one, facilitated diffusion. If you notice, these yellow dots cannot go through these phospholipids. They need help or someone to facilitate their movement. Now, this is still passive transport, though. Don't get confused on that. Look, I got a lot outside, very low. See the arrow? It tells you which way we're going. So if I'm moving from high to low, it is definitely passive transport. But since I'm using a protein, I am now facilitated diffusion. So particles that need facilitated diffusion, there's basically anything that has a charge or anything that's a little bit bigger. So remember, if I'm fat-based, I can just hop in there through the cell membrane anywhere I want to. If I have a charge, I need help from a protein. So now let's go to osmosis. If you look at your picture, you're going to look and be like, wow, that's just like facilitated diffusion. You're right. It is. It still needs a protein to go through. It's still going from high to low, okay? The whole point of this one is water has a charge. So if you're like water, you have to use a protein to assist you to go from high to low. So if you look at osmosis, the key parts on osmosis is it is the movement of water. That is a big part, and it must go through a membrane. But remember, water always runs downhill, so it is passive transport. Unfortunately, this causes cells in the human body some problems, okay? Like this. If we look over here, this is the problems that osmosis does for us. Water always moves in, and water always moves out. We can't stop it. The whole point is we really want to try to make it so our cells stay the same shape. So in this one, this is called isotonic. These are forms of osmosis. Whatever water particles come in, water particles will go out, and my cells stay normal and healthy. If we get out of homeostasis and we have too much water in our tissues or too much water in our cells, it causes some chaos for us. So let's look at hypotonic. In hypotonic, more water comes rushing into the cell than leaves the cell. And remember, water's just going downhill. So think of you sitting down in a little, like, I don't know, ditch, and all of a sudden this water's coming down. You can be bucketing water out, but there's so much water coming in, you can't get it out fast enough, and the swell swells and swells and swells, and eventually the cell will pop. The easiest way to remember hypotonic is, remember, hypotonics turn into hippos. So if you ever hear anything about cells swelling, cells enlarging, cells turning into hippos, hippos are hypos. Now that sounds really scary because the cell eventually can burst if you don't stop that gradient of water. In contrast, we can go to this other side. In hypertonic, more water is leaving the cell than coming in. So if you take your cytoplasm and for some reason it's going outside into the body tissues, your cell is going to shrivel and shrivel and shrivel and shrivel. And if you've learned one thing in anatomy, you better have learned this. If you change your shape, you change your function. So shriveled cells in hypertonic don't perform very well and can be very detrimental to us as well. So when we talk about osmosis, yeah, osmosis is the movement of water through a membrane from high to low. The trick is there's different forms of osmosis. If my cell swells, that's called hypotonic. If my cell shrinks, that's called hypertonic. These are all forms of osmosis because you can't stop it. Remember, this is passive transport. So let's go down here. You're like, why don't I just stand here and catch that rock? You can't. That rock's going to run you over. All right, so we've done diffusion. We've done facilitated diffusion. We've done osmosis. Now let's talk about dialysis. Dialysis is a process that people go through when they have renal failure. Now, you should hopefully know renal from your med-term class, but renal means kidney failure. So the way that it works is they run your blood through a machine. Now, the machine is active, but the process of cleaning is passive. The coolest part is this blue tube right here. This is a dialysis tubing. If you can see, it's like a cool little strainer. Now, these are different particles. You can see these are angstroms. These are just size. So if you want to think of it this way, this is 160 millimeters. This hole is only 24 millimeters. There's no way that green ball is going to go through that little hole. But these guys can, because check out your yellow dot. He's only 5 millimeters, and your salt, which is less than 1. And these are just units of measurement. This is how dialysis works. You run your blood through the machine, and your red blood cells and your white blood cells and platelets are too big. So they go through the machine and go right back into your body. But the toxins and the wastes and the salts all can go through. So even though dialysis is a machine that's plugged into the wall, the process is actually considered passive because it goes from high to low. This is a great example of what we call a selectively permeable membrane. We talked about that with our cell membrane, a semi or selectively permeable membrane. Some things go through, some things don't. Filtration, I don't really teach too often. Filtration is another form of passive transport. It does get vital the more you study more in-depth anatomy, but filtration is generally pressure-based. So in this case, these blue particles did go from high to low, and there was this blood pressure that actually pushed them into the surrounding tissues. Now, they did not expend any energy to get out there. Now, the heart is pushing the blood, but in this case, the particles just got pushed out by pressure, and we call that filtration. All right, now let's switch gears to active transport. Now, when we talk about active transport, we talked about a couple things. Remember, you're pushing the ball uphill. So let's take a peek. I only see 3 guys out here, and I see tons in here. So your mind says, well, these guys should leave, but look at the arrow. I'm trying to make it even more crowded. There's only one way that I can go from low to high. I must use energy of some sort. So this is active transport when I go from low to high. Or if you ever see a yellow lightning bolt, that is active transport. Now, I warned you, we don't want to move small things, right? This is moving one red dot. Most of the time when we do active transport, we're moving big things, okay? Like I always think of Costco ranch. So when you guys go to Costco and you get your ranch on, okay, that's what I think of when you go to Costco and get your ranch. If I'm going to spend energy to move stuff, I'm moving a ton. So that's what we want to think of. So now let's go through our parts and pieces. So let's go and start here with endocytosis. I can't tell you how many times I've had kids miss these questions because they don't follow the arrows. So if we look, this says inside the cell. So obviously this is outside the cell. So this would be your extracellular fluid, intracellular fluid, or cytoplasm. And if you notice, look at the arrow going in. If I'm going into the cell, that's endocytosis. If you're going out of the cell, that is exocytosis. Now notice, you do not see ATP or energy anywhere here. But I am telling you, if your membrane is folding, it is active transport. Okay, if your membrane is folding, it's always active transport. If the membrane's a solid line and things are going through it, then you have to see if I'm going high to low or low to high. But if the membrane's folded, you're always assuming active. Now, these are all forms of endocytosis: phagocytosis, pinocytosis, and receptor mediated endocytosis. And it really depends on what you're taking in. And that's it. So let's look. Definitely endocytosis. I'm outside, I'm inside, I'm taking stuff in. If I take in a solid filled particle like food, that's called phagocytosis. Phage or phag means to eat. If I take in fluid, if this bubble is full of fluid, and there's other stuff in there too, but if it tends to be fluid-based medium, that is called pinocytosis, which is to drink. Now, the cells do pinocytosis more than any of the others. So every day a cell does pino and phagocytosis all day, every day. But the problem is, I really don't know what's in that food. The problem is, I really don't know what those green dots are. Sooner or later, the cell is going to struggle and need something very specific. If you remember in our last video, we talked about amino acids, and there's 20 of them. What if I'm missing amino acid like 7? I got to get it. And I'm not sure if it's in this bubble. I'm not sure if it's in that food. So the third one we have is the almighty receptor mediated endocytosis, which I'm always going to say from now on RME. In RME, the receptors on the cell membrane get involved. And once those receptors get involved, they grab something highly specific, things that you definitely need. Now, cells don't use RME very often. They only use RME when they need something highly specific that Pino and Phago are not giving them. And the last form of active transport is the almighty exocytosis. Now, if you look at it, this picture looks exactly the same as endocytosis, but you have to follow your arrows. In this case, I have all this product that either I don't want or I don't need. Follow your arrow, and it is going out of the cell. Remember, the membrane's folding, so that's active transport, and I'm moving a ton, so that's exocytosis. And congratulations, you have now finished the difference between passive and active transport. So do me a favor, there's 2 slides of reviews today. So knock out these 2 slides of review. This is from an old test I gave a long time ago. So then the numbering's weird. Just do the best you can on your numbering. So go ahead and knock this one out. Okay, if you need to, pause it. I'm going to the next picture. Then let me move my face. Go ahead and knock this one out. And then the answer keys are on the next page. So I hope you guys enjoyed this, and I hope you come back to learn as much as you can.